Good afternoon, everyone. My name is Katrina Chapman. Today is Tuesday, May 16th, 2023. Today we are going to be discussing chemoreception, the sense of smell and taste. Chemoreception is a process by which organisms respond to chemical stimuli in their environments. The chemoreception that allows us to distinguish odors is olfaction, and the chemoreception that allows us to distinguish taste is gustation. Olfaction transduction begins through your nasal cavity, through your olfactory epithelium. On the roof of your nasal cavity, you have about a 3 centimeter squared patch that has olfactory neurons. These neurons are connected to your cranial nerve, which allows for stimuli to be transferred to your olfactory bulb, processed through the olfactory tract, and then interpreted in your olfactory cortex. From there, it is sent to the cerebral cortex, which is responsible for population coding that leads to the perception of the vast variety of smells that humans can differentiate. Olfactory neurons are G-protein linked membrane receptors. The limbic system, the pathway from your olfactory cortex to your limbic system, involves your amygdala and your hippocampus. Here you have emotional associations for smells that are triggered and allow you to remember specific memories associated with those specific smells. Through gustation, we have 5 different pathways that are triggered. We have our sweet, which is associated with nutritious food. Sour, which is triggered by acid. Umami, also associated with nutritious food, but directly linked to glutamate and can be found in food products that have high levels of MSG. You also have your salty sense of taste, which is triggered by sodium, and then bitter, which is triggered by toxicity. One taste bud is equal to 150 taste receptor cells. Taste buds contain 4 distinct cell types. Type 1 cells are glia-like support cells. Type 2 cells are receptor cells, which we can see in this image here. You have umami, bitter, and sweet cells. Type 3 cells are presynaptic cells. They're also known as taste receptor cells, and this involves your sour cell type. Type 4 cells are your basal cells. Something I wanted to point out is that your type 2 cell receptors activate gustussin. Gustussin allows for taste distinction, which differentiates your sweet, sour, umami, salty, bitter. It distinguishes the different pathways that all of these stimuli are going to follow. It's a G protein that activates the multiple signal pathways. One of these pathways involves kokumi, which is Japanese for rich taste, and another pathway is capsaicin, which involves that spicy sense of taste. The salty and sour sense of taste are associated with ion channels because sour is acidic and salt is associated with sodium, both ions. The sweet, bitter, and umami are receptors that are involved in the activation of your G protein coupled receptors. Taste transduction starts through your oral cavity. You have your taste receptor cells that release chemical signals that will travel down to the gustatory neurons. These neurons follow the pathways of your cranial nerves 7, 9, and 10 and carry all of your stimuli information to your medulla. From your medulla, it's transmitted to the thalamus, where it's transmitted to the insula or your gustatory cortex. There, everything is processed centrally and distinguished through your population coding. This is going to break down the differences between the sense of smell, taste, and trigger a psychological or behavioral response that will lead to a specified hunger in your digestive system. Here you see my references for this slide deck. Now I would like you to take a few moments to review the worksheet that I'm going to hand out. If you review these slides, just let me know which slide you would like me to go back to, and I will travel back to that slide. If you have any questions, go ahead and ask. And everything you need to know to fill out your worksheet is in these slides. Here's a quick preview of what your handout should look like. Really simple, straightforward. You can use your textbook as referenced here and these slides.